Right, guys. So before we move further in the topic, I think this protein is a really large and a huge topic. So just before we go further, I think we'll just revise or review whatever we learned through. Just we'll do just a basic summary of it. Right. So I will just remove all this from here. So it will be like a quick recap with just main points, so that will just help us to remember this thing. Right, so before beginning for the protein structure and function, right, we are studying about the protein. So what is protein? So so the main thing is proteins are polymers of amino acids, right? So, and the amino acids are covalently linked in the characteristic linear chain, right? And there are different kinds of protein, that is enzymes, hormones, antibodies, lens proteins of the fiber, eye, or like feathers, spider webs, all different kinds of protein. And the enzymes are the most specialized protein. And amino acids, so there are different kinds of amino acids. There is common and uncommon. So I will just write quickly common and uncommon amino acid. So for common, like there are 20 amino acid from which all the proteins, most of all the proteins are created, and the structure of amino acid is like that's a alpha. It's also known as alpha amino acid. That is has and amine group, carboxylic group, side chain, and hydrogen. So all this common has this side chain and this amine and the carboxylic group, except glycine, right? So only the glycine has, instead of this side chain, it has another hydrogen atom. All right, and this 20 amino acids are how they are described with the three-letter abbreviation. Like for alanine is like ALA and one letter symbol, correct? And this 20 amino acids are divided into 5 groups. That is, first is non-polar aliphatic R group, then aromatic R groups, then polar uncharged R groups, positively charged R group, and negatively charged R groups. For the uncommon amino acids, so the uncommon amino acids are formed from the amino acid residues, right? And they are like that. For example, the amino acid derived from proline is 4-hydroxy proline, right? And there is few more that we study, right? So 4-hydroxyproline, and then there is like 5-hydroxyproline, 5-hydroxy lysine that is derived from lysine. Then there is like 6-N-methyl lysine, right? So there is so on different kinds of uncommon amino acids as well that are important for that has an important role in the biosynthesis or in, right? Then after that there is a peptide bond. So what are peptide bond? When two amino acids join together by covalent bond, that bond is known as peptide bond, and the chain that forms is polypeptide, right? And the peptide bond formation is an example of condensation reaction. Why condensation reaction? Because water is expelled from the reaction. Right? Then some proteins have, beside the amino acid group, they have another chemical components as well, and that proteins are known as conjugated proteins, and that group is known as prosthetic group, right? So conjugated proteins means having another chemical component. And for example, that is, what are the example? Lipoprotein, right? Oops. Glycoprotein, right? Phosphoprotein. So these are the example of conjugated protein, right? And so we just, right, we saw the amino acid, then we saw peptide bond. What is peptide bond? And different kind of conjugated protein. Then now the primary. So we also saw, we just saw the names of the different types of method that separates protein, that are like affinity chromatography or electrophoresis. and then we moved on to the protein structure, or rather the protein primary structure, what we discussed in primary structure. Primary structure, that's just a description of all covalent bond, mainly peptide and disulfide bonds, linking amino acid residues in a polypeptide chain. That is its primary structure, and mostly that's important is the primary structure is the sequence of amino acid. Right? We studied the sequence of amino acid. So we saw different methods for the sequence of amino acids. If you remember, there is in 1953 there was two major discoveries. The one was regarding the DNA, James D. Watson and Francis Crick, that deduced the double helical structure of DNA. But regarding it, in that same year, there is Frederick Sanger. In 1953 worked out the sequence of amino acid in the polypeptide chains of hormone insulin. Right? So what was the method that used Frederick Sanger to determine the sequence of hormone insulin? What he did, so for example, this is I will just write polypeptide or peptide. This is a peptide chain, for example. First determine the content of, or rather first determine the amino terminal amino acid. So what's on the amino terminal amino acid, how they will determine? By reacting with FDNB, that is 1-fluoro 2,4-dinitro right? For using this, they will determine amino terminal, or he will rather determine the amino terminal, what's on the amino terminal, or there is another agents known as densyl chloride or like diamyl chloride. So after determining, for example, there is a glycine on the amino terminal, so that after that they will determine the amino acid contained. So determine amino acid content by hydrolysis. All right, after that what will happen? Then we'll select a splitting agent or breaking the bonds agent based on the presence of target amino acids in the protein. Then after we found the contents of the amino acid, then we'll select a splitting agent based on the presence of target amino acid. Then after that, after we broke the peptide chain into a smaller peptide, right, after splitting agent, what we'll do, we'll just split into this peptide chain, is split peptide into smaller peptide chains, and by, for example, by using trypsin as a breaking agent, and then we'll sequence each small peptide chain, right? And after sequencing this whole peptide chain, we will determine the order, like which comes first and which comes second. Right, this was the procedure for Frederick Sanger. There was another chemical sequencing that was developed by Per Edman, right? Remember, and that's known also as an Edman degradation process. Right? So what happens in this degradation procedure? So the peptide bond, right? So I'll write it down here. The peptide chain rather is first, right? So in this Edman degradation procedure, so what? First, the peptide chain is reacted for only just at the amino terminal and leaving all the other peptide bonds beside this just intact, and this first will be reacted under a alkaline condition, that is high pH, with phenyl isothiocyanate, right? Cyanide, right? And then this, after this reaction under Mendeleev, which converts the amino terminal amino acid to phenyl thiocarbamyl, right? Carbamyl, or it is also known as PTC adduct, right? And then the peptide bond next to, and after, so this will convert, so I will write it down here, converts amino acid to this derivative, right? Amino acid derivative to convert this phenyl thiocarbamyl. Then what we'll do, the peptide bond next to this, so we converted first amino acid, and then the next amino acid beside this PTC adduct will be splitted in a step carried out in, that will be in the acidic condition, that is low pH value, that is bi-anhydrous trifluoroacetic acid, right? Trifluoroacetic acid with the removal of amino acid as. So after reacting the second amino acid beside the PTC adduct in the low pH with trifluoroacetic acid, that will remove the amino acid as, okay, amino acid as thiazolinone, right? Thiazolinone derivative, right? So this will be this derivative, and then the this derivatized amino acid is extracted. Then this derivative amino acid is extracted in the organics with organic solvent, and then converted to the more stable state. Extracted for more stable state, right? And which is the more stable state, that is phenylthiohydrantoin, right? This derivative phenylthiohydrantoin by treatment with aqueous acid, and after converting into this derivative, then this is identified that what is this amino, that this use of sequential reaction carried under the first basic and under the acidic condition provides a means of controlling the entire process, right? And the process is repeated until typically as many as 40 sequential amino acids residues are identified. So this is the Friedrich-Sanger process, this is the Edmund degradation process. After these two processes, we also saw a mass spectrometry as well to determine the amino acid, right? Mass spectrometry. So what is mass spectrometry? It was just a this is a tool just in chemistry that is used to determine the mass of the molecule. The molecules to be analyzed are referred to as an analytes, and first ionized in the vacuum, and then they are introduced into the electric or magnetic field, and their paths and the field of function of their mass to charge. So what it determine? The mass to charge ratio. So that's the mass for the molecule. So this method is used to find the amino acid sequence. So for this mass spectrometry, if you remember, there were two techniques were developed in 1988. Two methods were developed, or two techniques were developed. So the first was, so for this mass spectrometry, it's not applied to the macromolecules when this technique was developed, because this treatment was done in the gas phase, and if it applies to the macromolecules, it causes rapid decomposition. So in 1988, they developed two methods to determine the mass of macromolecule. The first was the matrix assisted, that is MALDI, MS, that is metric assisted laser desorption ionization mass spectrometry, right? So in this, protein placed in the light absorbing matrix, and with a short pulse of laser light, the proteins are ionized and then desorbed from the matrix into the vacuum. So that's why this procedure is mass absorption, laser desorption, ionization. And so this was the first method, and what was the second method? In second, the macromolecules in a solution are first forced directly through the liquid to the gas phase. The solution, what it does, it's a solution of analytes is passed through a charged needle that is kept at a high electrical potential, disposing the solution into a fine micro droplets. Then the solvent surrounding macromolecules rapidly evaporates, leaving the multiply charged macromolecules and in the gas phase, and that's why this technique is known as electrospray ionization. All right, mass spectrometry, MS, or also known as IMS, ESIMS. And for this mass spectrometry, like they use this mass spectrometry for the sequences, short stretches of polypeptide to determine the sequence of short polypeptide, and the procedure or the information expected for the technique that is used is called tandem mass spectrometry. And why it is called tandem mass spectrometry? What is the procedure for tandem MS, if you remember? So there is right. So the first, the protein to be identified is treated with the protease or any chemical reagent to hydrolyze it to the mixture of shorter peptide. Let's have a write it down. So protein hydrolyzed with protease or any other chemical agent, right? Then the mixture is put it into the injection. Then, right, there will be first one, then there will be a vacuum chamber, then there will be a second mass spectrometry, and then will be the detection. So what happens after this hydrolyzes? The protein solution is injected into this mass spectrometer. Here, first one, and in this box, the different peptides are sorted so that only one type is selected for the further analysis. So the selected peptide is further fragmented in a chamber between the two mass. This is the vacuum chamber by the collision cell. So what happens in this vacuum chamber? That is the collision cell. The peptide is further fragmented by high energy impacted with the collision gas, that is helium or argon, and that is bled into this vacuum chamber, and then this is transferred to this mass spectrometry 2, where all the peptide that is broken is only in one place, and then in this mass spectrometry 2 there, and in this second mass spectrometry 2 that measures the MZ ratio of all the charge fragment that are broken and transferred here. All right, and this, so whatever the amino acids are broken here is brought and the mass is detected of it, and from that mass the sequence is detected. How it sequence? Only the ambiguities are leucine and high isoleucine because they have only the same mass. Right, so this was the sequence of amino acids, how it is determined, and after that we studied about just an overview of protein structure, that is how the rotation is defined and how it is done by interaction, what type of interaction, that is hydrophobic effect that we see, like it can be forming a solvation layer, the water will be forming a solvation layer, and the hydrophobic effect will contribute to the weak interaction to the protein stability. Then there will be a weak interaction between the hydrogen to hydrogen bonds, then there is ionic interaction, and then there was van der Waal interaction, and lastly we studied about the protein planes, right, the Ramachandran plot, right, and we studied lastly right now about the Ramachandran plot and how the rotation will be presenting from the rigid and the planar surfaces of the covalent bond or the peptide bonds. All right. Yeah, so that was the protein structure that we studied till, and in the next video we'll study protein secondary structure, how that, if you remember, we studied about the alpha helices, how these alpha helices are formed. Right, thank you guys for watching, and don't forget to subscribe the channel. Thank you.